Hello, my name is Leah Dean, and today's date is April 16th, 2025. And today I'll be demonstrating a DNA extraction lab using split peas while explaining the scientific principles behind each step. So to start, deoxyribonucleic acid, or DNA, is hereditary material found in all living organisms. It contains instructions needed for an organism's growth, development, reproduction, and function. Every cell in an organism contains a complete copy of DNA, which is stored in the cell's nucleus, and it encodes the genes that determine physical and biochemical traits. I've soaked the split peas in distilled water overnight previously, to soften the tissues and I also transferred them into a mortar. And when I did that, I added a saltwater solution made by dissolving 2 packets of salt water into half a cup of distilled water. And you can see that here. And here I have, a cup, and we're going to filter the blended pea mixture through a cheesecloth into a glass. We'll go ahead and do that. And we'll discard the solids. And to our mixture, this is our pea mixture, we're going to add half a droplet of detergent. And I'm going to stir that gently with my glass stirring rod. And the detergent breaks apart cell membranes, which are made of lipids, and this step frees DNA from both the cell and the nucleus and helps separate it from other cellular components. And now we're going to add this mixture to the refrigerator for 10 minutes. Okay, next I will add our mixture to this test tube very carefully. And now I'll add the papain powder. And this breaks down proteins, including the histones that DNA is wrapped around, and it helps clean the sample and release more free DNA. And then I'm going to stir that gently with the stirring rod. Try and get most of that powder down there to actually mix it in. Just want to stir that gently so you don't disturb any DNA. Okay, and now I'm going to add ice cold 50% isopropyl alcohol to the solution by slowly pouring it at a 45-degree angle to form a separate layer. And DNA is not soluble to cold alcohol, so it precipitates at the interface between pea extract and alcohol. And the cold temperature helps slow down the enzymatic degradation and enhances the visibility, of the DNA. Oops. Just get a separate layer going. So we've got equal parts solution, equal parts alcohol. Thought about even I need a little bit more than that. Okay. Looks like we have some DNA forming at the boundary between the alcohol and the extract. I don't know if you guys can tell, but it's white, which is nice. That's kind of what we were aiming for. We've got stringy DNA forming all throughout there. It looks like threads or like mucus, and the amount is more than I expected. It's cloudy. You know, it kind of clumps together well. Very nice. Okay, so this confirms a successful extraction, and this simple DNA extraction demonstrates how basic reagents, salt, detergent, enzymes, and alcohol can break open cells and isolate genetic material. And this hands-on procedure reflects techniques used in labs around the world for research, diagnostics, and forensic science. So that's pretty cool. That's exciting. Thank you guys so much for watching.